The speaker already replied that they are ready. So let's go ahead with Suranga L. and from University of Limerick, Ireland. Over to you.
Okay.
I hope you can see my slides.
Yes, speaker, we are able to see.
Hello, everyone. Good afternoon here from Dublin, Ireland. Let me just first of all say that it's fantastic to be able to contribute to this global effort in some small way towards the common goal of tackling this COVID-19 crisis. What I hope to do in my presentation is not so much to show any primary research data, but rather to highlight some key insights from studies and publications on SARS-CoV-2 and other related coronaviruses, and how that might relate to this narrative of what I would call the hyperinflammatory state of COVID-19. So I will also frame the discussion around two thought questions which could form the basis for further discussions after the presentation. So by now we all know that age is the most important prognostic factor predictive of COVID-19 associated morbidity and mortality. I'm highlighting a couple of graphs here from notable studies which clearly demonstrate that increasing age is able to differentiate between severe versus mild disease and survivors versus non-survivors. But age is a pretty crude phenotypic marker, and it will be extremely important to try and understand the underlying pathophysiology responsible for this wide spectrum of clinical presentations that has come to characterize COVID-19. Here on the left, I'm trying to list some of these from asymptomatic mild to symptomatic moderate and severe, still associated with self-limiting disease and recovery, and then severe disease coupled with acute respiratory distress syndrome, multi-organ dysfunction syndrome, or in other words, a hyperinflammatory state. And down here, this relatively newly characterized entity of pediatric inflammatory multisystem syndrome temporally associated with SARS-CoV-2 infection, or PIMS-TS, which is empirically observed weeks after a child initially may have contracted the virus. So this wide variability of clinical presentation, at least in my opinion, begs the question whether we are dealing with a single entity or different specific subsets of disease under the umbrella syndrome that we call COVID-19. And in fact, other researchers have asked a similar or the same question, like Siddiqui and Mehra here, that you might be familiar with, who have proposed this three-stage classification system based on increasing degrees of severity. This is largely based on data from the Chinese CDC report of over 72,000 initial cases of COVID-19. And what's interesting to note, first of all, is this apparently biphasic nature for this syndrome. Initially, there may be what's called a viral response phase, which captures about 80% of cases associated with asymptomatic or mild disease that is self-resolving. Unfortunately, it's the about 20% of cases that go on into this subsequent second phase of the syndrome, or a post-inflammatory response phase, which is associated with the severe morbidity and mortality that we have seen around the world. Now there may be also within these phases at least 3 different stages of disease, depending on severity, based on specific clinical symptoms and signs, which are being gradually standardized across the world. And so this kind of epidemiology and based on models like this that have been proposed, it leads to my first thought question on whether we have enough, at least clinical evidence, to support a phasic nature for the natural history of COVID-19, or more specifically, a potentially biphasic nature of the syndrome. Taking a little step back, there is at least decent preclinical evidence to support such a notion. Some of that comes from significant work by Channa-Panavar et al. Back in 2016. First of all, it is well established that induction of the interferon 1 response and the subsequent innate immune response is absolutely critical to establish this important antiviral state when any host is challenged by any virus for that matter. And in a series of beautifully designed in vivo experiments using mouse-adapted SARS-CoV-1 viruses, again, the virus that was responsible for the SARS outbreak back in 2002-2003, they quite convincingly demonstrate a few key findings. First of all, when there is early interferon signaling and a robust response thereof, we can expect rapid reduction of viral titers and mild clinical disease. Similarly, and interestingly, with no interferon 1 signaling or minimal signaling, we can also expect mild clinical disease, but predominantly due to avoidance of this significant lung immunopathology mediated by the pathological inflammatory host response. Problems really arise in this middle scenario here, when there is initial rapid viral replication followed by a delayed interferon 1 response that is potentially overcompensating, and this results in the recruitment of inflammatory monocyte macrophages that orchestrate this severe lung immunopathology characterized by ARDS that we have observed. Now we know the genetic sequence of SARS-CoV-1 and SARS-CoV-2 are very similar, around 80%, so it is very reasonable to infer some of these findings to try and understand the pathophysiology of the latest human pandemic coronavirus. But in fact, now we have timely work by Blanco Myeloid et al., which was only recently published in the journal Cell, as you may be familiar with, in an attempt to map the transcriptional response involving a series of in vitro, in vivo, and ex vivo models. They also recapitulated and corroborated essentially some of the same observations, in that SARS-CoV-2 infection induces low levels of interferon 1 and 3 associated with only moderate interferon stimulated gene or ISG responses. Juxtaposed to that are highly elevated levels of cytokines and chemokines, particularly interleukin-6, and these conspire together collectively to cause this viral evasion of the initial host innate immune defenses associated with an exuberant cytokine production, which are the defining hallmarks of severe COVID-19 pathophysiology. What's really interesting is that both studies corroborated findings in one particular dimension, in that this pathological host inflammatory response is dominated by innate immune cells, namely monocytes, macrophages, and neutrophils. In other words, an imbalanced myeloid response or a myeloid signature is apparent here. I'm sharing you with this nice illustration with you by Moore and June in their perspective in trying to understand how this imbalanced myeloid response might be contributing to SARS-CoV-2 and the ensuing hyperinflammatory state. As we saw, this delayed interferon 1 response upon infection with a human coronavirus may lead to a late activation of macrophages, dendritic cells, and monocytes, which secrete a number of cytokines, particularly interleukin-6, that is capable of activating immune cells through cyst signaling and a variety of other cell types like endothelial cells via trans signaling, instigating a signal transduction pathway through the JAK-STAT signaling cascade, culminating in the systemic production of numerous cytokines, which eventually contribute to this hyperinflammatory state and the pathology of severe COVID-19. And so if that model were true, we would expect, or one can expect, that targeting this myeloid response may be of some clinical benefit in severe COVID-19 at least. And indeed, now we have some tantalizing early clinical evidence to support such a notion. I'm highlighting this study by Roshevsky et al. In Science Immunology, recently published, in which they first of all hypothesized that these macrophages may be contributing to the hyperinflammatory state by phagocytic uptake of viral single-stranded RNA. And in alveolar macrophages, these are detected by TLR7 or 8 with downstream activation of the Bruton's tyrosine kinase or BTK, and subsequently NF-kappa B, which switches on a number of genes, resulting in the secretion of a plethora of cytokines, including interleukin-6. And these in turn recruit more innate immune cells, contributing to this cytokine storm and a feed-forward loop, thereby bringing on this hyperinflammatory state. And so in their study, they studied prospective observational study. They treated 19 hospitalized patients with severe COVID-19 with the specific BTK inhibitor acalabrutinib. And what you see are on the right are panels that clearly demonstrate improving oxygen uptake efficiency and absolute lymphocyte counts over the course of acalabrutinib therapy, about 14 days, with decreasing C-reactive protein levels. And so what has been proposed is that the improved outcomes seen in these patients, especially in the supplemental oxygen cohort demonstrated here by the red line, is that the BTK inhibitor may be targeting these pathological monocytes and macrophages, thereby dampening this cytokine storm. And this evidence has been convincing enough to start a randomized control trial evaluating the efficacy of BTK inhibitors in COVID-19 patients. And so that leads to my second thought question, in that how can this immune response implicated in the initial mild presentation and later severe disease seen in COVID-19 be explained perhaps at a molecular level? Again, I believe we have to go a few years back to work on the pathobiology of related coronaviruses. I'm highlighting here a couple of studies by Kindler et al. And Deng et al. In 2017. And collectively, what they demonstrated was that, very interestingly, endoribonuclease function residing in the non-structural protein 15 or NSP15 of the coronavirus replicase transcriptase complex, or the RPC, can selectively cleave viral RNAs, thereby simultaneously prevent the activation of host cell double-stranded RNA sensors. Very importantly, like MDA5 being inhibited shuts down interferon 1 induction and expression of interferon-stimulated genes in an indirect, therefore suppressing this indirect antiviral effect. And similarly, inhibition of OAS RNase L and PKR can inhibit RNA degradation receptors, or PRR, thereby inducing a robust interferon response. In true coronaviral infection, the endoribonuclease is able to selectively cleave off these uridine sequences, thereby eliminating that PAMP signal and suppressing the interferon response. So here we see a plausible mechanism how the coronavirus is able to go undetected from the desirable initial innate immune response in the host, therefore allowing the virus to gain a foothold. So now that's all preclinical data from models which need to be validated, but here I'm attempting to summarize them and put these disjointed pieces of evidence into a simplified model for the development of COVID-19 and the ensuing hyperinflammatory state. So we can suggest that early induction of a robust interferon-1 response can lead to rapid viral clearance, translating into asymptomatic or mild disease. And similarly, if there is reduced or minimal interferon-1 response, this individual may still be rescued from the deleterious effects of the pathological host innate immune response, translating to symptomatic or moderate disease. Problems arise when there is this delayed interferon-1 response, as we saw in the preclinical models of SARS and SARS-CoV-2, resulting in the immunopathology and severe, sometimes lethal disease. Here I'm again trying to highlight this potentially biphasic nature of this syndrome, in that about 80% of people will fall into this initial viral response phase associated with self-limiting disease. Unfortunately, it's the up to 20% of cases that seem to progress into this subsequent post-inflammatory phase associated with severe morbidity and mortality. Now, if such a biphasic model of COVID-19 holds true and is validated, it might also, I believe, allow us to contemplate strategies to optimize existing therapeutics. And accordingly, we have proposed recently what we call the double hit hypothesis, in which two unique elements of the coronaviral replicase transcriptase complex are simultaneously targeted to optimize treatment strategies. Now, in this model, what we suggested was that the first hit may actually involve a backbone antiviral, such as a nucleoside analog like remdesivir, for example, which may be indicated in all stages of severity of COVID-19, but in particular, the initial stage when viral replication is dominant. Now, however, that first hit might not be enough, especially in cases when patients progress to that second stage where the host inflammatory response dominates, and a second hit might be necessary. Now, some evidence for that to be aware of from the recovery trial in UK, when they demonstrated a clear mortality benefit using the immunosuppressive steroid dexamethasone, but only in patients being either ventilated or requiring oxygen support. So I think there is some tantalizing early evidence to support a double hit hypothesis to try and optimize therapeutic strategies for COVID-19. And finally, I will also just point out one potentially experimental strategy of how that second hit might come about. In my reading of the literature here, I came across this study by Bharadwaj et al. Over a decade ago. Now, again, they used a series of beautifully designed in vitro experiments using the coronavirus non-structural protein 15 or endoribonuclease. Hopefully by now I have convinced you that the endoribonuclease is a critical enzyme for the coronavirus to gain a foothold in the host. I'm sharing a figure here for the purpose of reference only, but not going into too much detail. What they demonstrated was that modified derivatives of a prototype 16 nucleotide RNA sequence can act as competitive inhibitors of this endoribonuclease. Now, the prototype sequence was efficiently cleaved by the enzyme, while the modified derivatives were not cleaved. but still retained their affinity binding capabilities. So I think this is far from any immediate translational capabilities, but it gives a potentially novel strategy that needs to be optimized in further in vitro and in vivo studies. So I started off this presentation by aiming to talk about the hyperinflammatory state of COVID-19, but in the process of constructing the talk, what I recognized was that it is perhaps a little bit more nuanced than that. I conclude that there is emerging evidence for at least a biphasic course for COVID-19 with an initial viral response phase and a subsequent host inflammatory response phase. Seemingly, it is viral evasion of the host innate immunity that is a major precipitant of this hyperinflammatory state in up to 20% of cases. And there is good preclinical evidence. It is, in fact, the endoribonuclease responsible for cleavage of viral RNAs, thereby avoiding detection by cellular pattern recognition receptors. This, in turn, coupled with a delayed exuberant interferon 1 type response, is responsible for this pathological inflammation that we see in severe COVID-19. What you see on the right is Janus, the Greek god of doors and duality, as a symbol of this double-edged sword of this pathological inflammation, in that when it is early and quick, it is protective, but late inflammation is actually detrimental. And very interestingly, this pathological inflammatory reaction bears uncanny resemblances to the macrophage activation syndrome that needs further evaluation. And finally, I will finish off by saying that if the biphasic model for the COVID-19 syndrome holds true and is validated, it may also allow us to contemplate therapeutic strategies with a double hit approach, concurrently targeting aspects of the virus and the host to optimize treatments against COVID-19. With that, I will thank you for your attention and also thank all the researchers and healthcare providers tirelessly working everywhere, and importantly, the patients who make these insights possible. Thank you.
He's concluding.
Thank you so much, speaker, for the wonderful presentation. So before we proceed, I would ask if anyone wants to ask any question, if there are any questions for Suranga, if there is anything.